That's fine. I'm going to turn now over to David Muddyman, who will introduce our next presentation. Over to you, Dave. Thanks.
Oh, okay. Thank you, David. So it's first of all, I'd like to thank SciEx for being a strong supporter of US HUPO for many years, and especially Christy Hunter. So this afternoon, it's a pleasure to introduce Daniel Hornburg, who is a senior scientist at Seer, a life science company in California that focuses on precision health. So Daniel?
Yeah, thank you, David, and welcome to my talk. Let me share my screen. Here we go. And started. Okay, yeah, so thanks for this introduction. I'm a senior scientist at Seer, and we develop cutting-edge proteomic solutions for looking deep into the plasma proteome. So also have a look at our poster. But in today's talk, I would like to focus on a collaboration effort together with Christy Hunter from SciEx, where we benchmark and optimize scanning swath, which is a new acquisition mode currently developed by SciEx. And it's still in beta phase, so this is pretty new and pretty recent data. So all is work in progress, but I think it's a good opportunity to share it with the community. And I also actually really like the format. It opens the HUPO to a much broader audience, and I hope we transfer some of the experiences we have now into future meetings. So I have split my talks into two sections. I will get started by talking a little bit about the swath in general and the concepts of scanning swath to give you a better understanding on where we actually see the benefits and why we benefit from this new acquisition mode. And then we will look into a benchmarking data set, look at different readouts to assess performance and see where we gain and why that is the case. So let's get started with talking about swath in general, because I think most of you probably know how swath works, but just as a wrap up, as a reminder, in swath, of course, we try to explore the mass over charge range, which is where the peptides are eluting over time. So we have these two dimensions depicted here, the M/Z range, and then the retention time. And what we do is we split that range, that M/Z range, in pretty separate windows, usually between 5 and maybe 20 M/Z, and we isolate all the precursors at any given moment and subject them to fragmentation to generate complex fragmentation spectra. And this is done step by step by step by step. So it looks like that. We basically step through the mass over charge range across the retention time. And of course, we can now define certain characteristics of such an analysis. So we have the cycle time, which essentially describes the time from one stepping procedure to another. And this is, of course, important. We need to match that to the, for example, we need to know how fast certain peptides elute, because we want to make sure that we have enough data points across an eluting peptide. So that has to be tuned based on your flow rate and based on your gradient. And it can be tuned by either changing the number of steps we are doing, as well as taking into consideration the accumulation time and also the step width. So let's first talk about the accumulation time, which is essentially how much time do we spend at each given moment. And of course, the longer we spend on that acquiring data, the better we can determine like what is the signal and what is noise. So that gives us sensitivity. And also when we choose to take very large steps, we can more quickly go through the mass over charge range. We can cover a wider range, or we can essentially accumulate longer for the same range. The problem with having very large windows, obviously, is that you add complexity to your fragmentation spectra, which is why that can compromise then the specificity. In ideal case, you would have a very small window which only isolates a few precursors, so that it's very that the complexity is pretty low, but then you don't step fast enough through the gradient through the M/Z range. So there's always this balance to be met. In scanning swath, we do it conceptually different. Instead of stepping step by step through the M/Z range with very distinct windows, we slide the quadrupole. We slide with a quadrupole across that dimension. So we really scan the entire range. And this leads to a different type of readout, right? I mean, in concept, we still have very, very tiny steps here, but the difference is that now instead of having almost no overlap, we now have a really large overlap. And why is that conceptually different? Well, first of all, it comes with a different definition of, for example, cycle time, which is now depending on the scan speed, and the accumulation time, which depends on the overlaps of the window, as well as how fast we are stepping through the range. But the more important part of that is that we now can add specificity, because we get a better understanding of what actually happened, where certain fragments are coming from which precursor. So in order to understand that, let's quickly talk about specificity in general. So where do we get that from a swath analysis? So first of all, we have, of course, a time dimension, which basically tells us where in the chromatogram a certain feature eludes. Then we have a fragment M/Z per Q filter. That basically means we have a certain area we isolate, and we know that the precursor has to be somewhere in that area, which links back to the complexity question and challenge I raised before. And in scanning swath, we add a fourth dimension, which is an accurate match. Oh, I forgot to mention the intensity, of course, of the fragments. This is why good libraries help us in identifications, because the relative intensity difference of the B and Y ions can be used to make an identification. So in scanning swath, we add a fourth dimension, which is an accurate parent ion mass and fragment mass correlation. And for people that worked a lot with DDA, they probably instantly understand why that is helpful, right? In DDA, what you have one precursor and you subject it to fragmentation. And this is actually already a lot of information that you gain from knowing the exact correlation of a precursor to its fragment. For example, if you take a complex proteome, a human proteome, and you have a high resolution precursor, on average, you can have 5 to 10 peptides matching to that exact mass. So your MS2 actually can be pretty, it doesn't need to add a lot of information, right? So in swath, conceptually, we always have a much more complex MS2 spectra, which is why getting a correlation between the precursor ion mass and the fragments is helping a lot. And how is that done? Well, we will see in a few seconds how we move this quad dimension, this isolation window, across these different precursors. And just keep in mind and look at what happens in the spectra that will be shown down here as soon as the precursor enters the quad window and as soon as it leaves the quad window. Since we are sliding across it, we have a correlation and start to see B and Y ions as soon as the precursor window enters, as soon as the quadrupole enters the precursor area, and they disappear again as soon as the quadrupole leaves a certain area again. And that allows us to link precursor masses to fragments. And in fact, it can also help us to, when we have the same B or Y ion mass from different precursors, to link that to the respective precursor. So in other words, let's look at, for example, this MS1 spectrum here. We have a couple of peptides in it. We would generate in a swath window this type of fragmentation pattern here. And I've already marked here the peptide 1 and 2 with blue and red. But conceptually, it would be hard to really understand which of the ions is coming from what type of precursor, especially since the retention time dimension doesn't really show a difference. But since we add the quadrupole dimension in that case, we clearly see that they separate here, and we can assign that in the scoring model in order to better understand what fragments belong to which type of precursors, and also the intensity distribution. If we would have the same fragment, we would be able to assign that to a particular peptide. With this, I would like to switch to the second part of my talk, which is about looking at the performance and what do we actually gain by using scanning swath in a proteomics sample analysis. And before I get into the real data, I'd like to remind everyone, and this might be obvious for most of you, but I think it's a very important concept. In proteomics, we like to talk about IDs a lot because that's something that shines and can be solved pretty well. But there are, of course, other performance criteria like the accuracy of a measurement, the precision, and last but not least, the speed, especially if we go for population-wide studies. We want to process hundreds, if not thousands or tens of thousands of samples. So this is a pretty important aspect too. The problem with that, or the challenge, is that all these parameters depend on each other. So if I start pulling into the direction of speed, I might actually compromise the level of ID, or I might compromise the precision. So it's kind of like an elastic tetrahedron of which we want to optimize the volume in order to get the best possible performance. But in the end, it depends on the study needs and has to be reevaluated whenever you want to benchmark a workflow. It's good to monitor all of these and then prioritize for your needs what is the most important feature you want to push for. So a way to do that, and in previous presentations, people alluded to similar approaches, is to use spiking experiments that allow us to differentiate certain expected levels of proteomes based on, for example, mixing different species. We used one of that as a flavor of these spiking experiments, mixing plasma proteome with E. coli, with a E. coli proteome. And not only because we at Seer are interested in getting very deep plasma proteomes, but also because a plasma proteome, in comparison to, for example, an E. coli, offers a quite different dynamic range property. So in plasma, if we, for example, plot the intensity distribution of high intense up here versus low intense proteins down here, the distribution is pretty skewed. So you have a few proteins that make up a majority of the proteome mass, whereas in an E. coli or also a tissue sample, the distribution looks a little bit different, maybe like that, right? So, sorry, so this is actually giving us different challenges in terms of doing a deep analysis of that sample. And for that reason, we used this type of spiking experiment because it allows us, while diluting in a background of a plasma in a E. coli proteome and vice versa, to look at different complexity properties of these proteomes and to assess precision along the replicates, accuracy approximated by looking at the linearity of the response, and of course the number of IDs as a function of different complexity properties. So we started out by first of all looking at some of the basic parameters, and this is still ongoing work, but we started to optimize certain aspects of the scanning swath procedure, and I just want to highlight a few of those. We used the TripleTOF 6600 plus system, both for variable window swath acquisition as well as for scanning swath acquisition. Also keep in mind, variable window swath acquisition has been optimized for the last couple of years, and so there's a pretty high benchmark to beat. Another point I want to emphasize here, I think that's generally always important to mention, is when we talk about the length of the gradient and what does it actually mean, right? So there are actually two measures. One can talk about the length of the gradient, defining it by where peptides start to elute until the time where peptides don't elute or the wash starts, or you talk about the sample to sample time that it needs, that basically your acquisition takes. While the second part here is the more honest way, in my opinion, to talk about. In my talk right now, I will focus on this definition of the gradient length, just because we haven't had any time to spend, we didn't spend really time on optimizing the overhead that we still have. For example, when we talk about the 5-minute gradient, we basically talk from peptides are seen to we wash basically everything out, which still has a pretty large overhead that we're currently trying to reduce. And there are many different strategies to do that, of course, but we haven't spent time on that. So whenever I talk about gradient, keep in mind what we are talking about in real. So we played around with different cycle times, especially for the scanning swath, saw different performance behaviors for that. Scan binning, this is actually quite interesting aspect that also highlights like how the R&D process is developing. So the scan binning is basically the time, the detector, the frequency with which a detector actually accumulates a signal, and usually it gets binned into four packages. Every 25 picoseconds get binned together, but scanning swath produces so much data that we actually had a problem downstreaming it, and we had to compile it into packages of 12 to avoid some dropouts of the data. Everything was then analyzed using the 1-omics SciEx platform. Also, the DIA-NN neural net platform allows us to process that files, but the data that I'm showing here was processed together with Christy on the OneOmics platform. So here you see the gradients, 45 minutes, 20 minutes, 10 minutes, and 5 minutes. And just want to highlight here how much we actually really squeezed together all the features, loading the same amount, roughly 3 micrograms onto the system. And I would say like the 5 minute almost looks like a washout. So that was a pretty challenging sample to scan through, given the complexity of the sample too. And let's jump to some of the first results here. And here I plotted the gradient lengths versus the number of peptide IDs for both E. coli and plasma peptides identified in the respective samples. And we see that scanning swath really pretty much outperforms the variable window swath for any given window here, both for the E. coli as well as for the plasma peptides or proteome. Of course, in the plasma, we have less features, especially since we mix both together, which makes the sample even worse, right? And another take-home message is that the biggest win in terms of relative differences we see for scanning swath when we go down to 5 minute gradients, which is kind of expected because we just lose this specificity and therefore in the end also the sensitivity in the variable window approach. So another way to look at that is here in this bar plots, and what I want to highlight here is essentially we compared here now different isolation windows, 3 Thomson, 5 Thomson, and 7 Thomson, and you see like there's still room for improvement. We still need to play around with whether we can decrease it further to get even higher IDs. But what we see here is really that with what previously was a 20-minute gradient can now be done in 5 minutes using scanning swath. So now this is all ID, of course. Let's look into the more complex spike-in experiments where we now look into all the dilutions and see how the system responds towards looking at different proteome complexities. First of all, one of the things that obviously is different looking into either of the directions of the dilution is what the instrument will see. So the plasma matrix as a background kept constant, and a dilution of the E. coli peptides really changes how the TIC looks like. So we maintain these large spikes, which are probably peptides of the plasma, while the whole background is decreased by quite a lot. And quite in contrast, if we go the other direction, we basically reduce the large spikes, but the overall complexity stays the same. So this, I think, also emphasizes that with that approach, we can really try to understand the response and performance in the context of a changing complexity. So this is a pretty complex figure, but let me guide you step by step through the information that is depicted here. So we are looking here always at one sample as a column, and then I split it here, the identifications for E. coli peptides and plasma peptides in the respective sample, and plotted it here as a bar plot, all in triplicates. So we have these arrow bars here, and then this is basically indicating the dilution of, in this case, the plasma peptides. And what we see here is basically the same as we have seen in the very first pilot, where the scanning swath outperforms the variable window swath for the same window, for the same gradient length, and consistently gives high identifications in terms of peptides, while basically just spending 5 minutes on acquiring signals on peptides. What we also see here is that there's a little bit of a stepping. So as we decrease the complexity, we gain a little bit more in the E. coli peptides, while when reducing the plasma peptides, which is kind of expected, and actually even more prominent when we turn it around. So as soon as we start diluting out a complex proteome like E. coli, we really gain sensitivity in terms of the plasma peptides. That's hinting us to another optimization homework. Basically, we try to close that gap, of course, and now having the data and trying to understand why we lose here a little bit of the sensitivity. I mean, it is still also expected, of course, but this gives us the data to try to make that stepping here less pronounced. Another thing that actually indicates that we are still work in progress, as you see here, we are not reaching exactly the same performance as we would like to see for adding another 10 minutes to the gradient. So probably here we have to reduce some of the samples. But overall, the ratio of scanning swath and variable window swath holds true here. And those samples will, of course, always run back to back on the same machine so that the relative comparison is still a fair comparison. Now let's have a look in the same manner at the precision indicated as a coefficient of variation here. The first take-home message is that we have a pretty high precision overall. So the interquartile range is always or almost always below the 10% mark, and the median is somewhere around 5%, and basically almost independent of the dilution, which is good. It's a pretty stable signal. What we also see is that we have slightly better precision for the variable window swath. And for the setting that we ran here, that is not actually surprising, because if we look at the accumulation time per feature, on average for scanning swath, it's about 5 milliseconds, and for variable window swath, it's about the double time. So we spent a little bit more time in variable window swath here, and this is probably why we have slightly better precision. But overall, it's still like a very high precision, especially also when we compared it to other pipelines. The same is true also for going into the other direction. We see here is one small outlier, but I also want to note here that we didn't do any data normalization prior to this type of analysis. So that might be just like injecting 5% more, and then of course the CV will be off. So there's some normalization we can do, but we wanted to look at the raw data to really see what does the instrument do. And the second step is then what can we do in terms of data analysis. Another way to look at that data is to try to understand how many features do we get below a certain CV threshold. I think those types of considerations are always useful, especially if you are in a clinical setup. You know your phenotype is very mild, and your sensitivity for seeing changes is very important. So you're not interested in high IDs that have a CV of whatever 30+%, right? So you are wondering, like, what do I gain, for example, below a CV of 10, and we can see that here in these curves. So overall, scanning swath, as expected, is better than the variable window swath. But what we see maybe as we look at the CVs below 5%, there we still have a small advantage for the variable window swath, at least for the longer gradients. For 5 minutes, not so much, because it's just not efficient enough to get us a specificity that we need to, in those short gradients, really capture enough features. The last part, I want to talk about the accuracy as approximated by the linearity of a response. So what I did here is taking the one-to-one mix of the mixed proteome and normalizing all the dilutions back to this one-to-one. So each of the peptides should basically was matched to its corresponding peptide in the one-to-one. So what we would expect is shown here on the right-hand side. The box plot should be always on the dotted line for the respective dilutions, and also the width of the box plot should be very small, which is a proxy for the precision, again, something that we have looked at in the previous slides. So here are some of the results. Again, like the gradient on the x-axis, and always the 3 dilution steps for scanning swath and variable window swath indicated here. The take-home message is that we have a pretty good accuracy or linearity for scanning swath, slightly better actually than for variable window swath. And here we probably just don't have enough features anymore to get a meaningful assessment of that linearity at a very high dilution for the variable window swath approach. But overall, it looks pretty good. The same, it's not entirely the same for the other dilution. It's still pretty good. And again, this is raw data. This is not normalized data, but we see we have an offset, at least for the short gradients, that we probably can normalize for. But in terms of seeing it on the raw data level helps you to understand where you have to move next. For the 20-minute gradient, it actually looks pretty good. So here we are really on the spot, especially for this lower dilutions, but even for the higher dilutions. And yeah, with that, I'm almost done. Just want to summarize that in this particular pilot experiment, we evaluated how we can benefit from scanning swath acquisition by using ultra short gradients, and we see that we gain on average gradient length, but in the end, the biggest percentage gain is for very short gradient, which opens up opportunities to go very fast and to have a much higher sample throughput. The next steps we are going to do in collaboration with SciEx is, of course, doing further optimization. Internally in the lab, we always talk about optimizing the box. As a concept, it's essentially knowing that the mass spec has a certain capacity it can work at, and what we want to make sure is that we use it throughout the gradient and the time spent on a sample as good as possible. So we want to get to this kind of box shape. If you just recall the TIC picture at the beginning, we are not yet there entirely. So this is certainly something where we can further benefit from, as well as optimizing all the different parameters that we need to set and can set in the context of scanning swaths, and of course reducing the overhead time, especially when we go with that short gradient. Every minute we save there is essentially like a big contributor to the overall runtime. And with that, I would like to thank you for the attention and for being here and participating in that experiment of doing research 24/7 essentially for people around the world. Special thanks to the Seer team, in particular Tahir, Patrick, Matt, John, and Charlie for their feedback on that research project, and to Christy, Hunter, Nick, and Stephen from SciEx. And if you're interested in how we get super deep plasma proteomes using the Seer technology, have a look at our poster. And with that, stay healthy and curious, and looking forward to your questions.
Okay, thank you, Daniel, very much for an outstanding presentation. I have a few questions from the audience members. So the first one is asking, for a low abundance peptide, how much better is scanning swath compared to regular swath? Because they think that the peptide in scanning swath would be less time spent on a peptide in scanning versus regular.
Yeah, that is true. So I can't give you like an exact number now, but it's certainly something that we have to quantify. So overall, we get better sensitivity by the fact that we don't need that much information, right? You don't need the cleanest MS2 spectra anymore, because you have this really tight correlation between the precursor and the fragments. So that's certainly helping, but completely true that it is a function of how much time you also spend on this particular precursor. And there's probably a sweet spot that we still need to titrate. And the sweet spot, unfortunately, it's always pretty complex. So it will probably depend on the sample itself, on the gradient again. So this is exactly what we're trying to figure out now.
Okay, thank you. So can another viewer would like to know if you could comment on how many proteins are being detected in plasma and E. coli for your 15-minute gradients, because you throughout your presentation you talked mostly about.
Yeah, it's all peptides. So the exact number of proteins, I think, for your the reason why we focus on peptide is that in the one omics cloud for now we don't have a protein FDR. So I think we had a couple of hundreds of proteins for the plasma proteome, but since we don't, the one omics cloud doesn't yet provide the protein FDR, it wouldn't be really fair to drop that number. That's why I focus on the peptides. For E. coli, I think it's definitely more than 1,000 proteins that we get for even the short gradients. But again, like since we don't have a protein FDR, it is something that we certainly are integrating, or not we, but SciEx team is working on that. Yeah, to get an exact number, we would of course need to know the FDR.
Okay, and one last question. Can you elaborate more on the scan binning and how that's related to cycle time?
Oh yeah. So the scan binning, and also Christy can jump in if I forget anything, but the reason, so the scan binning is basically done in order to aggregate signal, basically where the signal is detected. So every 25 picoseconds, there is a signal basically recorded, and then this is binned into, aggregated across four of these pieces, right? So, and this is slightly different in scanning swath now. So we aggregate more, we aggregate yet instead of 4 pieces, I think the current setting is 12 pieces that we aggregate. So it's not really affecting